My name's Brendan O'Connell. I'm in Ed Green's lab at UC Santa Cruz. I mostly work on de novo genome assembly, method development, sort of assembling new genomes for a species where we don't already have a reference genome for it. The technology I work on, the sort of methods development that I do, all of it can be applied to human genomes as well. And for example, cancer, a tumor, frequently is genetically kind of different looking from the patient that has that tumor. There will be these structural rearrangements in the genome where parts of the genome have been duplicated or deleted or switched around, jumbled around. It almost is like having a new organism sometimes, that there's so many changes have happened that if you sequence the tumor looking for, okay, what went wrong, what caused this cancer, how do we treat it, you have to treat it as though it's a new genome that you have to reassemble. So potentially, hopefully, what I'm working on has that application as well, that you can extend out something that works for a novel species will also work for a tumor that has a strange genome arrangement from what we're used to seeing. My grandfather was an engineer who worked on the Apollo program, and so I had sort of a exposure to engineering and science from an early age. And then actually in high school, at a career day talk, the guy came in who was an alumnus of my high school and was one of the early cancer immunotherapy researchers. He was looking at the use of monoclonal antibodies to suppress certain types of cancer. And the combination of him talking in my biology class with that AP biology class was like, I think I want to do this. So yeah, that was sort of what got me into it. So I'm maybe a little unusual in our lab in that I do both wet lab biology, so I'm in the lab actually pipetting things and doing work, and then also computational biology as well, so the analysis side of things. So I generally sort of split my time about 50-50 between those two. So I don't really have a usual day. I have sort of days where I do more wet lab work, days where I spend most of the day on the computer, frequently actually a month at a time where I'm coming into the lab and primarily running experiments all day long, and then I'll have a month where I run maybe 2 experiments in the lab the whole month, and then most of the time I'm working at my computer doing analysis. There's an organization that we actually just last week, the conference was held in Santa Cruz. That's the Genomes 10K conference, which is a group of scientists, including the PIs in this lab, who want to sequence 10,000 different species across all the vertebrates. There's sort of this exponential increase. Every time the Genomes 10K conference meets, it goes from like 1 or 2 genomes to 10 to 50 to 200 now. So it keeps getting faster, and there's new technologies, and even in the last year actually, there have been a number of new technologies that have finally matured enough to maybe start being really helpful in making this process faster and cheaper. And I think probably within the next 5 years, we'll see the actual physical sequencing cost, which used to be the prohibitive part of making a genome, is going to be much less prohibitive. It's going to be much cheaper and much easier. And it's really going to come down to more the just having someone sit down, collect the DNA, do the work. I like to make things. I like to design new things. And it's something that occasionally I'm driving home from work or I'm talking to someone else and I realize, wow, I'm actually doing really cutting edge research that I don't even think about it as being really cutting edge. I think about, yeah, this is what we're working on in the lab right now. And then I go, nobody else has done this. It's actually really the realization when I'm trying to find papers in the scientific literature and trying to find other examples of people doing the same thing so I can, reference that and get sort of a sense of how do I do this, that I go, oh, nobody's ever actually done this before. Well, that's pretty cool. I like that. That's a realization I have every few months, is working on something where I'm sort of in new territory, and it's hard, but it's a lot of fun. It's a good challenge to work on new research.